So here we're going to zoom in and take a look at oxidative phosphorylation. Now, oxidative phosphorylation occurs during the electron transport chain. So remember when we talked about the electron transport chain, there were those four complexes, complexes 1, 2, 3, and 4. And complexes 1, 2, and 4 were involved in proton pumping into the intermembrane space. Now, when we pumped protons into the intermembrane space, we created this electrochemical gradient because there's more protons within that intermembrane space, and there's less positive charge in that mitochondrial matrix. Now, oxidative phosphorylation and chemiosmosis is the process in which we utilize this electrochemical gradient to create ATP. So essentially what happens is that we have this protein, this enzyme, and it's referred to as ATP synthase. Now ATP synthase, it utilizes this electrochemical gradient. These H plus these hydrogen ions are going to get pumped into the mitochondrial matrix, and as they move into the mitochondrial matrix, ATP synthase is going to utilize this energy release to transfer a phosphate group onto ADP and yield ATP. Now, for every four hydrogen ions that move through ATP synthase, we're going to get one ATP. Now, the purpose of this video is to understand that this is oxidative phosphorylation and it's not substrate level phosphorylation. Remember that during glycolysis, we discussed substrate level phosphorylation, and that is the direct transfer of a phosphate group from a high energy compound to ATP. Remember that in glycolysis, we produced glyceraldehyde 3-phosphate and we produced phosphoenol pyruvate. These were two high energy molecules that contained phosphate, and once we hydrolyzed that phosphate group, this high yield reaction was able to transfer that phosphate group onto ADP to produce ATP. Whereas in oxidative phosphorylation, we are taking free inorganic phosphate. This inorganic phosphate is not bound to a high energy molecule. It simply just exists and it gets added on to the ADP to form ATP. So that's the difference between oxidative phosphorylation versus substrate level phosphorylation. In oxidative phosphorylation, we are dealing with free inorganic phosphate that gets added to ADP, whereas in substrate level phosphorylation, we had those high energy molecules that contain phosphate that get transferred, and that phosphate group gets transferred to an ADP molecule. Now, some key definitions to keep note of. Chemiosmosis itself is the movement of ions down their electrochemical gradient. So in this case, chemiosmosis is these H plus ions moving down their electrochemical gradient, and that energy release drives the reaction of ADP plus inorganic phosphate to yield ATP. And this part summarizes the electron transport chain itself. So the electrons are carried via electron carriers such as NADH and FADH2. Now, these electron carriers obviously pass on these electrons to carrier proteins that exist within the electron transport chain. Now, as these electrons move through the electron transport chain, hydrogen ions are pumped into the intermembrane space, and this is going to generate that electrochemical gradient, which is going to give the energy necessary for the proton. It generates the necessary energy for the proton motive force, which is going to drive ATP synthesis. So that is essentially oxidative phosphorylation. It's the conjunction of inorganic phosphate coming together with ADP to form ATP, and it differs from substrate level phosphorylation because in substrate level phosphorylation we have phosphate within a high energy molecule and it simply gets transferred to ADP.